Okay, good morning, everybody. We're going to continue in chapter 20. And here's where we left off in this chapter on carbohydrates in our last lecture. I just introduced these tables and want to remind you that you will have kind of the left-hand portion on your resources page on the exam. And so I will probably take off whether it is a ketohexose or a aldopentose, but you'll have the name and you'll have the structure of these monosaccharides. And we're about to do a practice problem with one. So this one says in the monosaccharide hemiacetal. So I want to encourage you, if that word in of itself, you don't have something come into your mind, a picture of a hemiacetal, pause and go to the end of chapter 15 and review a few of those slides to remind yourself what a hemiacetal is. Okay, it is a carbon that has a hydroxyl group. Now, it doesn't matter what group is pointing where. What matters is the pattern of bonds all attached to this carbon. It's got an alcohol hydroxyl group on it. It's got one ether type group. Okay, so this R is some sort of alkyl continuation. And then we have a hydrogen bonded on this carbon, hence the acetal part. It used to be an aldehyde. And then we have some sort of other carbon-based, put a little R prime, or put it on this side, other type of bond. So that's the definition of a hemiacetal. That's what we're looking at below. It says number all the carbon atoms and identify the anomeric carbon atom. Also identify it as either the alpha or beta anomer. So this part we covered in our last lecture. I would go up and find the slide on that if you've already forgotten what alpha and beta indicate and what they mean. So let's identify the carbon that is the hemiacetal. Or actually first, this is number. Okay, so we're going to number these carbons. So this is pretty easy to do in a straight chain form. In straight chain form, remember you kind of have all your carbons in a row like this, and you've turned the chain on its side. We've got 1, 2, 3, 4, 5. We would have 6 carbons here. But what we're looking at is the cyclic form. All right, so we want to think, because this is a 5-membered ring and not a 6-membered ring, that means the straight chain had a ketone group in it, okay, just like this. And where I'm pulling that information from is where we talked about the difference between pyranose and furanose, okay? Furanose is a 5-membered carbon ring. Pyranose is a 6-membered carbon ring, and often you get the furanose when you had a ketone group in your straight chain monosaccharide. So that's what we have, which is why we end up with a 5-membered ring. And so the carbon here, to number these, we will actually start numbering. This would be carbon 1, 2, 3, 4, 5, and 6. All right, so carbon, just to kind of give you an idea, carbon 1 and carbon 6 will not be included in the group because, I don't know if you remember, it is the last chiral carbon, the OH group on the last chiral carbon, farthest from our carbonyl carbon. Okay, here is our carbonyl carbon. And the last chiral carbon is carbon 5. The OH on carbon 5 is what will be used to close the ring, so we're closing it between a bond between this O and that carbon. So that means carbon 1 and carbon 6 will actually be excluded when we close the ring. So that's important when you number this. Wanted to take a second and acknowledge that. So this is actually carbon 1, which means we'll go up into the ring now. This will be carbon 2. This will be carbon 3, carbon 4. Here's carbon 5, and then just above, we know carbon 6 always sticks out above the ring. So it might have been easier for you to actually number in reverse because you always know this carbon 6 is above the plane of the ring. That might have been an easier way to start versus where I did with carbon 1. But either way, that's how you would number these carbons. Okay, identify the anomeric carbon atom. That's going to be this one right here, carbon 2. It is the only one that is bonded to, if I can kind of identify some of these groups for you. Okay, here would be carbon, oxygen, carbon. That's the ether bond. Okay, if I can change my highlight color, carbon with OH group. Okay, and then we have two carbon 2 carbon bonds off of this, carbon-based bonds off of this, which actually makes this exact carbon right here, carbon 2. This is the anomeric carbon. What I'm pausing and thinking about for a second, if you're wondering why I'm kind of sounding like I'm pausing for a second, it says monosaccharide hemiacetal. And technically, when you look at carbon 2 here, we don't have, it would be a hemiketal is what it would be. I think that might be a typo, because if you look at the other 2 bonds, maybe you can see green. We have a carbon bond here and a carbon-based bond here. We don't have a hydrogen on carbon 2. So this should be a whoops. Hemiketal, okay. But that is the anomeric carbon. We have the correct one labeled. And then, oh, is it the alpha or the beta anomer? So again, you want to look at the orientation of the OH group branched off the anomeric carbon. Since it's in the same plane as carbon 6, we would say it is the beta anomer. All right. Here's a challenge for you. I encourage you to go do this on your own. Identify all the chiral carbons in alpha D fructose, ribose, and deoxyribose. So what we have here, I believe, if we look up at our table above, yeah, we have fructose. So, this is fructose. So I'll go ahead and do fructose with you, and then you can also try the other ones. What color do I want to use? The chiral carbons. I'm going to erase some of my markings. We're going to go through and just identify chiral carbons here. So I'll highlight them in this color. 3 would be a chiral carbon. 4 would definitely be a chiral carbon. 5 would be a chiral carbon as well. And then 2 would be a chiral carbon. But 1 and 6 are both bonded to 2 hydrogens, so those would not be chiral carbons. So again, take a minute, go look at, we just did fructose, so that one's checked off, but go look at ribose and deoxyribose and see if you could do the same thing. All right, now the bulk of the lecture today will be talking about reactions with monosaccharides. So there's a few different kinds we'll look at. There will be a reaction section on exam 2. So pause this video as needed. Write down questions that you have for me, since we're not doing this in person and you can't ask in person. But we're going to start with the first one, which is going to seem somewhat familiar. It's just applied in a new context to monosaccharides. So this is a little bit of a review here. It says, recall, aldehydes can be oxidized into carboxylic acids. This is the abbreviation for an aldehyde. This is the abbreviation into a carboxylic acid. And I want to remind you where we saw that. We saw it in our chapter on aldehydes, which was chapter 15, right here. Okay, this right here is what we're talking about. We looked at this exact reaction. We talked about the oxidation of carbonyl carbons. So we have some sort of oxidizing agent that helps us do this. Remember, if we're oxidizing a molecule, we're going to increase the number of carbon-oxygen bonds, and that's what you see here as the aldehyde becomes the carboxylic acid. We have this new carbon-oxygen bond right here. And so think a monosaccharide has an aldehyde at the very end, and we're going to turn this into a carboxylic acid. So let's go look at how that's done. First off, we've got to convert it from its cyclic structure to the open chain form. This can only occur with open chain monosaccharides. So in its open chain form, let's highlight, here is the aldehyde. This is where the action is going to occur. And guess what? It is this particular hydrogen bond right here that we're going to break and convert it into an OH bond first. All right, but remember, at physiological pH in the body, where these monosaccharides are going to be, carboxylic acid groups actually exist as carboxylate anions. They lose their acidic hydrogen. And this hydrogen right here is our acidic hydrogen. So we're going to lose that. And when we do so, the carboxylic acid group looks like a carboxylate anion. Okay? So these carbohydrates, so for example, glucose here, we call them reducing sugars. Because they reduce the oxidizing agent, okay, that is used to increase the number of carbon-oxygen bonds. They are reducing the oxidizing agent while they themselves are being oxidized. So we call these reducing sugars. And the way you identify a reducing sugar, some monosaccharides are and some monosaccharides are not reducing sugars, is you want to check and say, do I have a hemiacetal carbon? You always want to look at this carbon right here and say, is it a hemiacetal? Do I have one hydroxyl group, one ether bond, a hydrogen, and a carbon bond on this carbon? If it is a hemiacetal, then yes, it's a reducing sugar. Okay, if it's an acetal or something besides a hemiacetal, it is not a reducing sugar. Okay, don't worry about this. I'm not going to go into this, but what I did want to talk about is this picture here that maybe some of you have noticed or been looking at. This particular reaction is actually a very important one in nutrition and just food in general. It's called, the browning that you see on this meat here happens, that type of browning on food happens because of this exact reaction, reducing sugars reacting with amino acids. And the name for kind of this, it's a series of reactions that occurs in food. And the name for this series is known as I'm probably going to botch how this is pronounced, but the Maillard, I always say it different ways. Maillard reactions. Okay, so in food, you can think about bread, how when we cook dough to make bread, if you bake bread or any baked good, you might get a browning on the outside of the bread as it bakes. As you sear meat, you see kind of the browning on the meat, and that is because the sugars in the food, any carbohydrate portion in the food, can react with the amino acids, and again, it's a series of steps that browns the food. That's why the aroma or the smell of coffee, bread, chocolate, again, other baked things, is due to this series of reactions. So reducing sugars with amino acids is how we kind of get that browning or that char on different foods. Okay, so that's one example of one reaction, simply oxidizing an aldehyde. So notice it's only monosaccharides that have aldehyde groups, okay? If you had a monosaccharide with a ketone group, I'm not talking about that. We're focusing mainly on the aldehyde into the carboxylic acid group. Next reaction is the reduction of aldehyde sugars. Okay, so again, here is our aldehyde group. What happens when you reduce an aldehyde? And I encourage you to actually pause and think through that before I just talk about it. That is, again, another, we actually saw it in chapter 14 and in chapter 15. Aldehydes are reduced into, you can probably read, an alcohol, okay? So here it's being reduced, so it's reacting with what we say a reducing agent, and it's forming an alcohol, so we call these sugar alcohols, okay? And here is glucose is our example. All that we're doing is we are taking these hydrogens and we're breaking this, kind of zoom in on this, we're breaking this carbon-oxygen double bond into a single bond. One of the hydrogens is going onto that oxygen, forming the OH group, and the other hydrogen is going directly onto the carbonyl carbon, increasing the number of carbon-hydrogen bonds. Remember, oxidation is increasing carbon-oxygen bonds. Reduction increases the number of carbon-hydrogen bonds and reduces the number of carbon-oxygen bonds, which is what happens here. We go down to one carbon-oxygen bond and we increase up to 2 carbon-hydrogen bonds. All right? So when this happens with monosaccharides, with sugars, and we call them sugar alcohols, we change the name ending to let a reader or a person know that it's no longer a regular monosaccharide, it's a sugar alcohol. Okay, so sugar alcohols are named as derivatives of sugars by replacing the name ending. Instead of glucose, it would be glucitol. Or, whoops, I didn't know why I marked that out. Glucitol, okay? So we changed the OSE ending, fructose, ribose, deoxyribose, glucose, to ITOL. So glucitol, also known as sorbitol. What's next, my slide that's next, is really a derivative of this bullet point. You have to be careful with sugar alcohols because they can give you very unwanted side effects, lead to some upset stomach down the road if you consume a lot of them. All right. And this is, people who have type 1 diabetes and have to really watch their glucose levels, to eat sweets and desserts will often revert to goods and consume foods with sugar alcohols. Sugar alcohols pass right through our digestive tract. They are not absorbed into the bloodstream because they're a different compound. They've been manipulated. And so that is why they can upset our digestive tract. And so this is always some comic relief. I don't ever read this in class, but this is a Amazon review on, this product over here, Haribo gummy bears, sugar-free gummy bears. The title of the review is pretty funny. 1-star review. So you can read this if you want. I will definitely give you a heads up. It's not exactly PG, okay? What I think is hilarious, though, is that over 13,000 people found this helpful. It's quite a story, okay? And you will definitely avoid sugar-free gummy bears, especially before exams, which is what this student found himself, the situation he found himself in. All right, so we've talked about oxidation and reduction of monosaccharides and how we form the carboxylic acid or how we reduce it down into a primary alcohol. Now we're going to talk about, this is kind of a preview, getting ready to talk about how monosaccharides link up into disaccharides or polysaccharides. So to prepare you for that, I'm going to read this right here. Hemiacetals react with alcohols to form acetals. Okay, so again, these are a lot of big words. This involves a lot of atoms. You want to read this and think about this slowly. Remind yourself what a hemiacetal is. Here's a picture right here. It's a carbon with an ether bond. There's that. Same carbon has a hydroxyl group. And to make it a hemiacetal, we also have a carbon-hydrogen bond, all on the same carbon. When we react this with an alcohol, here is some OH group, methanol, ethanol, any kind of alcohol, propanol. What we form is an acetal, which is a carbon with 2 ether groups bonded to the same carbon, and you can still identify a hydrogen directly bonded to that carbon. Okay, if there's no hydrogen on it, it might be a ketal versus an acetal. But when there's a hydrogen on it, we say it is an acetal. Okay, so there's the formal definition. And when we think about cyclic hemiacetals, okay, these are cyclic monosaccharides, such as glucose, other monosaccharides. When they react with alcohols, we call the product glycoside, okay, a glycoside. So we see a picture of glucose down here. What is going to happen, the focus kind of on the part in blue and red, we are going to break off the hydrogen and the OH from the alcohol, and these are going to come off to give us water. We always get water as a byproduct of this. And then the alkyl-based substituent is going to link up to the oxygen to give us a new carbon-oxygen bond, which is going to give us a second ether group. So it's turning this hemiacetal into an acetal. So again, if we highlight these groups over here, and I want to do this color. Here's carbon, oxygen, carbon. That part never changes. What's new is the new ether group right here. Boom, boom. Okay, so this is a glycosidic bond. I honestly don't like the way things are colored here because when we looked at, I think it's fine. I'm not going to go into that. I don't want to be any more confusing. So, you recognize, this right here is your glycosidic bond. Okay, so we've shown a very simple alcohol. Okay, but remember monosaccharides have all sorts of alcohol groups on them. So that's why 2 monosaccharides can react together to form a glycosidic bond and thus a disaccharide, which is what I'm going to show you in the next slide. Also want to point out acidic conditions. This needs a catalyst to react. So take note of those things. They might help tip you off on different reactions on the exam when you remember which reactions need catalysts, which reactions need strong acids, so on and so forth. Okay, this is a continuation of formation of a glycosidic bond. That's what we saw here. hemiacetal turning into an acetal, forming the glycosidic bond. Here we're just looking at it between two cyclic monosaccharides. All right, so again, focus on the part in red. We are going to break off. It doesn't matter really which monosaccharide you're breaking the OH off of and which one you're just breaking the hydrogen off of. What matters is that collectively you lose water. Okay, so if you're not sure what I just said, what I mean, let me kind of repeat this. It doesn't matter if you break this bond and this bond, okay, the OH off the first monosaccharide and just the hydrogen off the second monosaccharide, or if you break this bond and this bond. Either way, you would get the same thing, okay? It doesn't matter monosaccharide 1 or 2, which one loses an OH and which one loses an H. It's just that collectively you lose HOH, water. Okay, and when that happens, we link the two monosaccharides together into a disaccharide, okay? And these are held together by a glycosidic bond. Specifically, if you notice, this is an alpha glycosidic bond. Why is it called alpha? Well, over here we had since the OH is oriented down, opposite of the plane of carbon-6, the CH2OH, this is an alpha monomer. This also would be an alpha monomer. Now, it doesn't matter if they're both alpha, but what matters is the orientation on your first monosaccharide of that OH group off of carbon 1. Okay, this is carbon 1, 2, 3, 4, and 5. So the OH on carbon 1 is oriented opposite the plane of carbon 6. So this is an alpha monomer. So when you form the bond, what is very important is that the orientation of this is still angled down. That's going to be very important. We're going to do a carbohydrate lab, dry lab, and when you draw these bonds, you want to think about how the OHs on your rings were oriented. You want to keep them the same, and when you link monosaccharides together, you want to keep their glycosidic bond angles the same, okay? Notice, too, the OH on your second monomer, okay, which is carbon 1, 2, 3, 4. The OH on carbon 4 is also, guess what, down. So that's why over here, let me erase, the angles of both of these are down. Down and down. That's very important, okay? Now, when you have this downward angle, forming a downward angle glycosidic bond, we call it an alpha glycosidic bond. Okay, that's very, very different from, I don't know if it's going to show another example. I actually want to scroll down to find another example. I'm going to come back to hydrolysis in a second. That's going to be very different from, here's a good example, a beta 1,4 glycosidic bond. Okay, beta, the orientation is up because the monomer, well, it's not showing monomer here, but the monomer here, the first monomer, must have been beta, and so we want to maintain that orientation when we draw the glycosidic bond, okay? So take note of what these little, in the blue circles, what those bond angles are. We'll come back to all the numbering and everything in just a minute. And you can get some wild ones over here. This one looks very different, okay? So we'll talk about that one in a little bit. All right, let me go back up. Okay, so this is joining two monosaccharides together. We lose water when we do that. We form a glycosidic bond. This is how you form disaccharides. If we just kept going, like adding on more monosaccharides in this direction until you get this big long chain, you could form a polysaccharide is what we'd call it at that point. But the second half of this slide, the bottom half, says hydrolysis of a disaccharide. So guess what? We're just literally going to take, if you look at the top half of the screen, we're going to run this in reverse, and that's hydrolysis. So forward reaction would be the formation of a disaccharide, and the reverse of this, which is what we see on the bottom, is the hydrolysis of a disaccharide. And it's literally just the reverse of the top half, where we have our disaccharide using water. We're going to break water into H and OH, okay? We're going to break this ether bond on these carbons, and we're going to break the monomers in two, and then we're going to restore the hydrogen would go back on this oxygen, and the OH would go back on this carbon, okay? to restore our 2 monosaccharides. Okay, that's what you see over here. Again, it doesn't matter whether you break, let me zoom into this side, or whether you break this side. All right, I think actually in the structure, the product, they broke the bond on the left side of the oxygen, but it doesn't matter whether you break the bond on the right or the left. You're going to put the pieces of water back on to restore your monosaccharides, and you should end up with 2 hydroxyl groups back on those carbons. Okay, so again, that is hydrolysis. To remind you, hydrolysis, we're using water to break bonds, and that's what we're doing here as we take these monosaccharides, convert them down into, or say, take this disaccharide, convert it down into a monosaccharide. And this is what happens as we eat food, carbohydrates that have simple sugars like sucrose, or a long carbohydrate, long polysaccharides. Water is used in our digestive tract to start breaking it down into individual monomers that are all then converted into glucose, that are then pushed into cells so we can run glycolysis and make ATP. So the body is doing this, hydrolyzing all these little glycosidic bonds. We've got to have the correct enzymes to do this. So that's why some starches are digestible and some are not. But that's what our body's doing when we digest carbs. Okay, practice. We're going to draw the structure, so make sure you do this with me. Maybe you want to pause this video and try it ahead of time, and then watch me go through it and see how you did. Or just do it along with me, either way. But we're going to draw the structure of the alpha and beta anomers that result from a reaction of methanol. So what is methanol? Try to draw that out before me. That is a 1-carbon alcohol. So you should have CH3OH, or you can draw the Lewis structure out if you want to, but there's methanol. And ribose. I've actually already drawn ribose. You can pause and go find ribose on a table up above and redraw that down onto here. And this is, are these compounds acetals or hemiacetals? All right. So I've only drawn the alpha, and I probably need to redraw the beta. Let me see if this will work. Copy. Will that work? Looky there. So I'm going to erase, to give you the beta as well, I'm going to erase the orientation of the hydroxyl group on carbon 1 and put it up. So this would be our alpha monomer. This would be our beta monomer. Again, because the orientation of the OH here is opposite of the CH2OH, carbon 6. And I guess it'd be carbon 1, 2, 3, 4, carbon 5 here. Only 5 carbons in ribose. And then, oh no. Here the beta, because it's oriented up, same plane as CH2OH. Now we're going to react this with methanol, CH3OH, so plus CH3OH, and we're going to draw the product. Okay, so we are going to break off. Let's see which bonds we're going to break. We're going to break off the hydrogen from the alcohol in both cases, and then we're going to break off the OH group on the monosaccharide. And we're going to get a direct new bond. I'm going to draw this in blue between this oxygen and this carbon. Now notice we're not breaking any bonds, other bonds in methanol. Just so wherever the O goes, this little CH3 is going to go with what was methanol. And this part that I'll highlight, the OH here and the hydrogen from methanol, this is going to give us water. So that's one of the first things we'll get is water. Okay. And then we'll redraw our monosaccharide. And we should now have, and we want to keep the orientation. So on alpha, I'm going to draw the angle, the first group down. And instead of OH, we broke that off. It'll now be OCH3. Okay, this is what was from methanol. Whoops. Okay, that was from methanol. And then you'd want to, of course, draw the rest of your OHs the correct way. Here for the beta, redraw your ring, keep the CH2OH up above. And this time, since it's beta, I'll have OCH3. There it is. Okay. Now, the question is, are these compounds acetals or hemiacetals? So we look at this. I'm going to go in and highlight a few things. Let's use that. Maybe purple is good. Here's the carbon I'm interested in. Okay, here's what I see. And let me scale this down. So we're going to have carbon, oxygen, carbon bond. There's one ether group. Second, carbon, oxygen, carbon bond. There's two ether groups. I only see 1, 2, 3, right? 1, 2, 3 bonds on this carbon. So what that means is there is a fourth one to hydrogen. Okay, so this is not a hemiacetal. This is definitely an acetal because of the two ether groups. Same idea down here. Highlight that if you can see it. This is an acetal. This is our beta monomer, and up here, this is our alpha monomer. Okay? All right, moving on. I'm not going to spend a lot of time on this next slide, but we can throw phosphate groups on monosaccharides. So let me read some of this. Phosphate esters contain, I wanted that to be red. Phosphate esters contain a phosphate group bonded to the oxygen atom from a hydroxyl group. Now notice, there are hydroxyl groups all over this thing. So which one are we going to put the phosphate group on? And the example I'm going to show you with glucose, and this happens in our body, the first thing we do when glucose is shoved out of the blood and into a cell is the cell phosphorylates. That's what we say when we put a phosphate group onto a monosaccharide, we phosphorylate the monosaccharide. And here, the phosphate group is actually going on carbon 6. It's going to go right on this oxygen right here. And we see that new phosphate group right there. It's linked up through the oxygen to carbon 6. Okay, that's going to help keep glucose in the cell so it doesn't like drift right back out of the cell so that we can then run glycolysis. So what's happening is hydroxyl groups on sugars bond with these phosphate groups and they form what we call a phosphate ester. These are products, reactants and products all throughout metabolism in glycolysis as shown below. We'll actually look at this exact step and this exact enzyme when we get to, I think it's like chapter 22. And anyways, I'm not going to spend a lot of time on this, but we can add phosphate groups. If you want to draw out the Lewis structure of what this looks like, I'm going to draw my little ring here so you can understand how the bonds are working out. Oops, it's a terrible hexagon there. But we have CH2 and then O, and then here's a giant, it actually changes the shape of this thing quite a bit because this phosphate group is really big. There's a lot of atoms involved. Okay, so these oxygens right here would have a negative charge on them. Anyways, now you have this giant phosphate, and it's phosphate ester because of this carbon-oxygen phosphorus pattern, okay, attached to your monosaccharide, and it prevents glucose, in this case, from drifting right back out of the cell. Keeps it in there so we can run glycolysis. Okay, so I think that's it for the reactions, I think. Oxidation, reduction, linking them together to form disaccharides, and hydrolysis. So 4 total that we went over. And I wanted to show you this table. This table always really intrigues me. It's classifying monosaccharides, disaccharides, right here. Sugar alcohols, and then synthetic sweeteners. You might recognize a lot of these in different foods you can buy or consumer products you can buy at grocery stores. And it's relating their sweetness, how they taste, how sweet they taste. Everything is relative to sucrose, which gets a rating of 100. Okay, so here's sucrose. This is, the sugar we would dump in chocolate chip cookies if we're making those. So what's interesting is fructose, the monosaccharide you find in fruit, is way sweeter than just sucrose by itself. Your sugar alcohols are about as sweet as sucrose. And then what's astounding is these synthetic sweeteners. Aspartame that you might find in like diet sodas. Sucralose. So I don't know if you've ever bought the little drops you can add to a bottle of water to kind of flavor your water. That will often have sucralose in it. 60,000 times sweeter than sucrose, which is why you only need like a drop of it in, say, a regular size water bottle. But still, I mean, these are wild how sweet they are. I see these two a little less, but aspartame and sucralose are pretty well known. And they're just insanely, their sweetness level is insanely high compared to our natural fructose, galactose, glucose, things like that. Lactose is milk sugar, not very sweet at all. Anyways, I always like to point those out because it's pretty astounding. Okay, now we're going to kind of dissect. We're going to go back to the idea of glycosidic bonds. I scrolled down to this slide earlier, but we're going to really look at the differences and the numbering that comes into play when you are labeling a glycosidic bond. Okay, so we're only going to do this for common disaccharides. There's probably a lot more options than that exist out there, bond formations, glycosidic bonds, than what we'll talk about, but we're just going to look at the most common ones. So bonds between disaccharides are formed as glycosidic bond between two monosaccharides. That makes sense. Now, the glycosidic bond can either be alpha or beta. We already talked about that, actually. The alpha bond extends below the ring or rings, and the beta bond extends above the ring, okay? Honestly, this is always with respect to the first or the monosaccharide on the left, okay? So I actually want to erase that S because we're really just considering the monosaccharide on the left. That would be this one and this one in each case. And its orientation with respect to that monosaccharide. So last point here, the glycosidic bonds create often, not always, but often what is called a 1,4 link because they usually, not always, but usually connect carbon 1 of one monosaccharide with carbon number 4 of the next monosaccharide. So again, you can see the numbering of each carbon in each hexagon monosaccharide structure. And we are linking carbon 1 with carbon 4, and the orientation is below the plane of the ring, which makes it an alpha 1,4 glycosidic bond. Or you call it an alpha 1,4 disaccharide. Okay? Scoot over here. We are linking again carbon 1 of that monosaccharide with carbon 4 of this monosaccharide. The arrow you can see orienting that glycosidic bond upwards from the plane of the ring leads us to call this a beta 1,4 glycosidic bond or beta 1,4 disaccharide. All right? And I'm going to show you the one I was alluding to earlier. This is actually the bond we see in sucrose. Okay? The most common disaccharide. And the way it links monosaccharides together is way different than what we just saw. It links carbon 1 of the first monosaccharide with carbon 2 of the second monosaccharide. So how we would say this, I don't know if it's 1, 2. How you would write that then, and this is definitely oriented downwards, right? So it would be an alpha 1, 2 glycosidic bond. So that one's kind of unique. I think there's some more pictures of these on these tables. So again, these are kind of showing you maltose is a very common disaccharide, lactose, very common disaccharide, alpha 1,4, beta 1,4, and there's sucrose, which is a beta 1,2 anomeric bond or glycosidic bond. What does this say over here? Notice, I'm reading this, each of the corresponding enzymes in an ACE. We'll get to that in chapter 19. I'm not too worried about you knowing enzymes right now. But this will be helpful when we talk about this in chapter 19, all the enzymes. Excuse me. Maltose is digested by maltase. Lactose is digested by, it's over here somewhere, lactase. Okay, so people that are lacking lactase, this is where you run into GI issues with dairy because you don't have the proper enzymes to digest these disaccharides. Also, before we move on, it says notice which disaccharides are reducing sugars and which are not. So again, you always want to look at, to identify that, you always want to look at the terminal, the last monosaccharide, and can you find a hemiacetal group on carbon 1, on the terminal monosaccharide? And it's actually pointed out here in maltose. So yes, it is, check, a reducing sugar. Again, you see the hemiacetal on lactose. So yes, it is a reducing sugar. But on sucrose, if you look at carbon number 1, I see an ether group, and then I see a second ether group. There's no OH group on this first carbon. There's no hemiacetal. This one is an acetal. And so this one is, you'll see, not a reducing sugar, because glucose is now an acetal. So, and ketal, we don't have it on the fructose monomer either. So, that actually leads us right into this practice question. What do we have left? Really not much left. We're almost done. Okay. Practice question. It says the disaccharide cellobiose can be obtained by enzyme catalyzed hydrolysis of cellulose, which is a really, really, really long chain polymer, carbohydrate polymer. Do you expect cellobiose to be a reducing or non-reducing sugar? So again, all we want to do is come over to the terminal monosaccharide. That's the one on the right. We want to look at carbon 1 here, and we say, is this carbon a hemiacetal or an acetal? I see the ester bond, the ether bond, and I see the OH, and then obviously to be a acetal or hemiacetal, we have that hydrogen there. This is a hemiacetal. So this would definitely be a reducing sugar. Then it says, how would you classify the link between the monosaccharides? So we want to zoom in on that part. I would number the carbons if I were you. We've already numbered carbon 1 on our monosaccharide on the right, so that would make this carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6. On the monosaccharide on the left, this would be carbon 1, carbon 2, 3, 4, 5, and 6. So this is a link between carbons 1 and 4. So we know it'll be either alpha or beta, but it's going to be a link between 1 and 4. Now we just got to decide, is it alpha or beta? And because the orientation of the bond is up above the plane of the ring, we started with a beta monomer of whatever monosaccharide this was. So it would be beta 1,4 glycosidic bond. So I might move that over here. This is beta 1,4. This says, show the structures and name the 2 monosaccharides that are formed upon hydrolysis of cellobiose. So if we hydrolyze this with water, H and OH, and we break this bond, and we put the OH back on this carbon, and we put the hydrogen back on that oxygen, what 2 monomers would we get? You might notice the orientation of our OH groups is down, up, down, and then also over here, down, up, and then this one will be down as well. So these are going to give you 2 identical monosaccharides. To compare that, to name that, since you don't have these structures memorized, I'm going to scroll back up for a second. We're looking for, on the hydroxyl groups 2, 3, and 4, down, up, down. So you go back up to these tables right here, and the monosaccharide that matches that on carbons 2, 3, and 4, if we number these 1, 2, 3, 4, you'll see glucose is down, up, down. So when you digest or hydrolyze cellobiose, you're going to get 2 identical monosaccharides of glucose. All right, and then the only thing we have to decide right here is whether those monosaccharides are alpha or beta. The first one, since this is oriented up, would be a beta glucose. They would both be beta glucose since this one's also oriented up. All right. Again, if I'm moving through this too quickly, you can pause and rewatch it as many times as you need. But if you still have questions, please write those down so that we can talk about those. This is just a table of some important polysaccharides. Okay. Notice this is based on glucose. So the first one is cellulose. These are just units linked together of glucose. Lots and lots of glucose units linked together. What's different about cellulose is that it is a beta-1,4 link. I don't know if you remember, last week I said we do not have the enzymes to digest cellulose because we don't have the enzymes to break apart a beta-1,4 glycosidic bond. We do have the enzymes to digest or break apart or hydrolyze alpha-1,4 linkages, which is why things like amylose, starch, are digestible to the human body. Okay, that's kind of indicated right here on the table. Here we can hydrose because we have this particular enzyme, alpha amylase, but we don't have a beta cellulase type enzyme. Amylopectin, also digestible to us because the linkage is alpha-1,4. And then glycogen, alpha-1,4, this is actually the stored form in the body of glucose. This is how we store glucose in our bodies, in our liver. So definitely digestible to us. All right, these blanks over here, amylose, amylopectin, both referred to as starch. They are both found in plants. Amylopectin has a branch point. That will be shown on the slide below, our very last slide. Glycogen and amylopectin, glycogen also has a branch point. Those are told here and here. Well, it's an alpha-1,4 link with an alpha-1,6 branch point. So I'm going to talk about that in a second. Those are the branch points. Carbon-6 is a branch point. We'll point that out in a second. Okay, glycogen and amylopectin look identical, but they're found in different sources. Glycogen is found in animals. Okay, they're found in us, in our liver. Amylopectin is stored form of starch in plants. All right, last slide here. These are polysaccharides, and this kind of figure out here, these little individual dots represent monomers of glucose, and so as we link glucose together, the branch points are helpful because it allows us to link more and more and more versus just one long chain that we would have to coil into some crazy structure to store. But because we have all these branch points, we can store a lot more glucose this way than we could otherwise. And then this is amylopectin, what it might look like in plants. So let's go look at the structures. Cellulose, I'm not going to talk about that. Glycogen, this is what I want to focus on. So you see here, it kind of has these unique formations on the ring structures. It kind of angles them in these different, what they're called chair conformations. But what I'm going to point out, if this highlighter will work, is this, you can always find the carbon-oxygen-carbon bond to kind of orient you. And then it's numbered. There's carbon 1 of one monosaccharide. Here's the other. Here's the glycosidic bond, alpha-1,4 link to carbon 4, so alpha-1,4. Now, if this is carbon number 4 on our middle, kind of this blue shaded ring here, then that makes this carbon 5, and then you already see this is labeled carbon 6. This is a branch point in glycogen. What that means is you can form a second ether bond on another monosaccharide to branch off carbon 6. Okay, so when we see these little pieces break off, these represent these little branch points, which is why, again, we can store lots more glucose in our liver due to that. Okay, so guess what? We could also, this would be carbon 1, 2, 3, 4, and 5, 6. We could branch off this carbon. Okay, this would be carbon 6. We could branch off this carbon. So if I ask you to go in and label any branch points on a disaccharide or trisaccharide, I would want you to label the carbon 6 as a branch point and even be able to draw in a branched monomer like this off carbon 6. It's just an ether bond up to carbon 1, and then you'd finish the ring up there. This will help after we do the carbohydrate lab. I think that's this week. But that concludes our lecture for today. I know that was a lot, but we have finished chapter 20. We will have a quiz online, so make sure you take care of that today as well. And see you guys in class Thursday.